Hello children. In the last modules we have discussed the various forces which helps the water to rise up in the ascent of sap. What are the various forces that helps in the water to rise up in so you know that the conductive tissues are there. So various forces, what are the various forces? This is what we were discussing. And here today let us discuss the importance of root hair in the upward movement of water. What is the role of root hair in the upward movement of water? And we will discuss some of the experiments also. In that you know that root hair is the extension of the epidermis. So definitely root hair is under soil, so definitely it is surrounded by the soil particles. So in the soil particle, in between the soil particle, it is porous. You know that soil particle, in between the soil particle there will be water. So this water can get into the plant, so get into the root hair by the process of osmosis. So as far as the water intake is considered, intake of water is considered, it gets into the gets into the root hair by the process of osmosis. So once the water entered into the root hair, then slowly it moves into the epidermis. From there the epidermis to the inner cells. The other day I have told you, we call it as cortex. Below the inside the epidermis, we can see, if you take the cross section of the root, you can see that there will be cortex, and cortex is followed by the endodermis. Hence it reaches the xylem. So here you can see the root hair is surrounded by the soil particle. In between that there will be water. That water, by the process of osmosis, enters into it, and as the water enters the root hair, then into the epidermis, then the remaining tissues, cortex, many layers of cortical cells will be there, and then it enters into the xylem. So xylem is the conductive tissue, or this is the vascular tissue. So xylem of the root, it is continuous with the xylem of the stem, xylem of the leaf, like that it continues. It's a continuous column. Okay, so here once the water is entered into the xylem by the process of osmosis, there's an upward push that push water upwards. Okay, we can call it as, that's due to the turgidity of the cells that is already absorbed water. Okay, so this pressure that push water upwards, but this pressure is not, we can call it as root pressure. This pressure is not enough to push water till the leaf. Okay, it can reach up to a certain height. So especially in the case of herbs, this is possible. In the case of big trees and all, the root pressure is not enough to bring the water till the leaf. Okay. Now this is as far as the water intake is concerned. Now mineral intake. Minerals are also taken in by the roots. You know that you have learned the process of active transport. So you know that the root hair is again surrounded by the soil particle. In that soil particle there will be minerals. So salts, these salts exist as absorbed as ions. So ions, you know that as far as the water is concerned, water is moving from the higher concentration to lower concentration by the process of osmosis. This we have seen. But as far as the minerals or salts or the ions are considered, you know that here in the cell sap, that is the inner content of the cell, inside the cell sap there will be higher concentration of these salts in the cell sap, which is there, the vacuole, which is there in the cell, which has got higher concentration of the ions. But outside it is lesser concentration of the ions, but still these mineral ions are taken in by the process of active transport. What is that? By utilizing energy, the forcibly these salts or these ions are taken in. And once the ions are taken in by using ATP, we have seen in the last class, by active transport by using ATP, adenosine triphosphate, once it is entered into the cell, into the root hair, then further it moves along with the water from the epidermis to the xylem, and further conduction, upward conduction is taking place. Upward conduction of water, or otherwise we can call it as ascent of sap. Ascent of sap, it is taking place through the xylem. So this is what, these are the two concepts that you have to study. The xylem, what's the use of xylem, upward conduction of this water and minerals, and phloem is the other conductive tissue or other vascular tissue, or you can say other conductive element. Okay, xylem and phloem. So phloem is involved in the downward conduction of the prepared food, glucose. Downward conduction, that's the purpose of, that's the function of phloem. You have learned it in the lower class. Now we have got few experiments to show the absorption of water, and here you know that root, you know from the lower class that the root absorbs water. Okay, here there are two experiments set up. Two test tubes have been taken, and both the test tube, this water, test tube number A as well as B, there is water, and both the test tubes few drops of oil is also kept, so that you can prevent in both the test tubes, so that you can prevent the rate of transpiration to a certain extent. Now, in the test tube number A, you've got a plant, small plant, herbaceous plant, small plant, and you keep this experiment set up for some time. You can keep it in a test tube stand or so, this experiment set up for some time, and you can observe that after some time, here the level of water goes down in the case of test tube A, whereas there is no movement, no change in the level of water in the case of test tube B. Why is it so? So the reason is root is absorbing water, here there is no root. The absorbed water is moving up through the plant, and as it reaches the aerial part, what is the aerial part? Stem, leaf, etc. are the aerial parts of a plant. So from the aerial part, transpiration takes place, so loss of water takes place, so that much of water to be replaced, okay, replenished by, that is absorbed by the root from this test tube. That is why you are getting the result observation that the water level in the test tube, it is going down. Now another experiment to show that, second experiment to show that water conducts upward through xylem. You all know that there are various plants, delicate plants, balsam plant, or any plant which delicate herb plant you can take. Okay, you yourself can conduct this experiment to see the conduction through xylem. Take a beaker or a small container, transparent container will be ideal, transparent some container will be ideal. Take water in that, take any kind of dye, okay, ink or blue ink, red ink or blue ink, or dyes. If you under the laboratory conditions, when you make, you can use eosin dye. Okay, so put few drops of dye into this beaker, and then what you have to do, you have to keep the plant in this water. Keep it for some time, and you observe that. If you observe after some day, maybe a day it will take, and if you observe, you can observe that inside the stem there's a red color. What is the reason for that? Then you take the cross section of the stem. How do you take the cross section of the stem? As you hold the stem just vertically, just facing your face in your eye level, and take a cross section. I'll show you once when you come to the lab. Okay, then cut it and view it under the microscope. You can see that this center region of that cross section, it is red in color if the red is taken as the dye, or blue in color if you have taken some blue color dye. Okay, so that shows that some cells which is present in the center, it is responsible for conduction of that water. Okay, so from this it's clear that we know that the cells that is present in the center is nothing but xylem. So conduction of upward conduction through xylem, it is taking place. That's what experiment number 2. So you have to learn this for the examination. You may be asked to write the aim of the experiment and how to write the inference, observation, etc. I'll show you once in the next module. I'll show you how to write the experiment. Okay. Now experiment number 3, here conduction, again to show the conduction through xylem. You have to take two experiment setup, 2 beakers. Okay, in the first beaker I have taken a small plant, and I need delicate plant I have taken, and this delicate plant is cut under water. So you've got beaker, inside that there's water, you can see the marked water level, and I need to cut the stem under the water by keeping it under the water. What is the reason? If I keep it outside and cut, what will happen? The air bubble will end, so I may not get the exact result what I am expecting. So I have to cut under the water. The same other setup also, the stem need to be cut under water. By keep, what do you mean underwater? Keep the stem under below water level and cut it. Cut it with the help of a razor blade. Okay, then there won't be any entry of air bubbles. Then I am keeping the stem in such a manner that with the help of I am holding it very well, and here you have to do one thing. You can see over here 3 centimeter length outer region of the stem is taken away. Okay, then around 3 centimeter, and you can find that the outer cells, the epidermis, cortex, cambium, phloem, everything you can see it's been removed. Only the center part is visible here. See the other case, in the beaker number B, you can find that center part is removed. Center part is removed. Outer part is kept intact. So the first condition we can call girdling experiment. You can call it as girdling experiment also. That is, outer region is removed. I have to keep maybe for 1 or 2 days. After 1 or 2 days, if I observe, I can find that here this plant where the outer region is removed of 3 centimeter length, you can observe that still those leaves which is there in the upper part remains turgid. Why? What is the reason? Whereas in the experiment setup B, you can find that where I have taken with the help of incision, I have made an incision there and I have removed the central tissues and the outer region is kept intact. There I can find that the leaves are drooping down. What is the reason? What could be the reason? Here, it is possible. In the case of setup A, it is possible to have the conduction of water through the xylem. Xylem is there in the center. Xylem is there, so conduction is possible. Whereas in the case of B, setup B, where the central part is removed, so no conduction is possible. That is why here the leaves are drooping down, whereas here the leaves remain intact. Now, the fourth experiment, I have taken a potted plant. Either I have to take a potted plant or maybe a small plant, or I can take a small twig of a tree. Okay, I am just taking, and there also I have to remove the outer tissues, epidermis, cortex, again the phloem I am removing. Okay, and I am keeping this setup, and upper region there will be leaves. So I am keeping the setup for some time, and I can find that the diameter of the region which is above the cut, okay, it is increasing in diameter. Okay, the region above the cut is increasing in diameter. Slowly it is increasing in diameter. Why is it so? And another observation which I can see is that here from the cut region some sap is coming out. So you can observe some sap that comes out from the cut region. From this region you can see the sap comes out. So what is the reason for this? And but the leaves remain intact. There is no drooping down of the leaves. But these two observations you can have. One, the stem is increasing, upper part of the cut region, stem is increasing in diameter, and the sap is oozing down from the upper part. So from this you can conclude that why there is the increase in the diameter in the upper region. This is because of the presence of extra availability of food in the upper part. Whereas in the lower part that food is not reaching. So that lower part can't grow beyond a limit. Okay. Why there is upper part there is more food availability. After photosynthesis, the food that is going through the phloem is not possible as it reaches here. So only the exudation of the sap from this region, and so only that excessive amount of nutrients or excessive amount of food prepared will help to increase the diameter of the stem in this region. So girdling also you can say, this shows that the conduction of prepared food takes place through phloem. To conclude this chapter, let me reinforce these 4 terms. One is ascent of sap is possible with the help of root pressure. What is root pressure? Pressure developed in the root due to the intake of water by osmosis. Okay, so osmosis takes place, helps the cells to have help the cells to develop the turgidity. Hence a pressure. This upward push is a push from the lower xylem, that is xylem in the root to upward. That is called root pressure. Now next one, it is capillary. What is capillary? You must have heard of capillary action. Okay, so you should know that the xylem cells are arranged. Xylem cells of the root, stem, leaf, it is arranged like a tube. Okay, vertically it is arranged like a tube. So the thinner the xylem elements are. Xylem elements, what are the different xylem elements? Xylem vessels, xylem tracheids, you must have learned in the lower class. The xylem elements acts as a continuous column. Vertically continuous column. The xylem elements acts as vertically continuous column. And the thinner the elements are, more possibility for the rise of water by capillary action. Upward rise of water by capillary action. The third thing is that transpiration pull. What is transpiration pull? Transpiration pull, you must have heard of this transpiration, that is loss of water as a result of from the aerial part of the plant as water vapor due to the heat of the sun, right? So here what happens, this loss of water create, so you know that as the water is lost, that water is to be replenished. So there is a suction force from above, suction force. Root pressure is a push, whereas transpiration pull is a pull, okay, from upward. Then the water is moving up, because the water molecules are bind to each other by cohesive force. Cohesive force means what? The force between the like molecules. Okay, so cohesive force, the water is rising up. That is called moving up. That is called transpiration pull. Next one, it is adhesion. What is adhesion? Adhesive force means the in this case unlike molecule, attraction between unlike molecule. The walls of the cells as well as tracheids and vessels, walls of the cells as well as the water molecule, there is an attractive force. So this attractive force will enable the water molecule to slide through the sides of the cell wall of these elements. Conductive elements. Okay, so this is called adhesion. So root pressure is not enough to push water upwards enough. So herbs it's possible. Transpiration pull is quite strong force, and capillary action that is enabled by the again transpiration also helps over here. As a result of transpiration creates a vacuum that will also help the capillary action. The transpiration also helps in the capillary action. Another one is adhesion. Adhesion is the attraction between the water molecule with that of the cells of the xylem elements. Okay, so all this will help in the ascent of sap. Hence the plants can have a healthy survival. Okay, so by this we are coming to the end of this chapter. Question answers are there. The one word question answers we will discuss during the our live class, and the other question answers I will give you as the next module. You are supposed to learn this chapter for the next live class. I will be asking question or conducting test. So for next live class, learn this chapter and come. Thank you.